Welcome. Today we have a really interesting talk. It's called Stress-Induced Beta Cell Early Senescence Confers Protection Against Type 1 Diabetes. And with us, we have Dr. Feyza Engin. She's a PhD and she's an associate professor at University of Wisconsin-Madison at the School of Medicine and Public Health. She received her PhD degree in genetics from Baylor College of Medicine and completed her postdoctoral studies at Harvard. Her initial studies using a pharmacological inhibitor of ER stress provided the first direct link between beta cell ER stress and type 1 diabetes in preclinical T1D models. Her lab generated the first genetic inducible deletion model of ER stress response and demonstrated that the loss of beta cell unfolded protein response, or UPR, sensor IR1 during early stages of disease prevents autoimmune disease in diabetes in NOD mice. Recently, Dr. Engin and her team demonstrated a previously unrecognized link between the UPR and senescence in beta cells during T1D development. And her current research focuses on beta cell stress, organelle dysfunction, and beta cell immune cell crosstalk in type 1 diabetes pathogenesis. She's the recipient of several prestigious awards, including NIH KO1 Award, a JDRF Career Development Award, and a Shaw Scientist Award, and she serves on the editorial board of Diabetes and Frontiers in Endocrinology. Hugo Lee, who is a graduate student, joined the Engin Lab in 2017 as a graduate student in the integrated program in biochemistry, and he defended his thesis earlier in November. So that, I suppose that makes him, he's a card-carrying PhD now. His main projects in the lab were focused on studying how disruption of different pathways of the UPR can be protective against T1D. Welcome, and thank you so much for joining us. I'm really excited to hear more about your work and share this with our larger global audience.
Thanks for having us. We're happy to be here. We sort of discussed this and thought maybe it would be better Hugo give a short presentation on the project. He was the lead author of this project, and then we can discuss it.
Sounds great.
Cool. Sounds good. Hi, everybody. Thanks for coming. So in the Engin Lab, we're interested in studying different types of elder stress in the context of type 1 diabetes and other autoimmune diseases. And today we'll be talking more about this recent paper that we just published this past month. So, okay, I'm sure to this audience, I'm sure this needs no introduction, and it's already well known that in mice, beta cells make up most of the islet, and that they secrete insulin in response to high blood glucose levels. But I just want to highlight that an important characteristic of beta cells is that they're highly secretory. So in this image, you can see and appreciate just how much insulin granules there are in one beta cell. And since they have to make, fold, and secrete a lot of insulin, their ER is under constant heavy load, and they're especially vulnerable to ER stress. And when ER stress is present, this pathway called unfolded protein response will be activated to try and resolve the stress. So ER stress activates the UPR, which has three main sensors. There's ATF6, IRE1, and PERK. They all have their own functions, but for the first part of our talk, we'll be talking about ATF6. So ATF6 is the transcription factor that's localized to the ER membrane. And when ER stress is present, it localizes to the Golgi, where it undergoes cleavage. This turns it into its active form, which can then move to the nucleus to activate and regulate the UPR target genes. So we use the NOD mouse model, which is a preclinical mouse model for type 1 diabetes. So to determine the role of ATF6 in type 1 diabetes, we made a beta cell knockout in our NOD mice, which we did by using the Cre-log system. So we made the ATF6 flux mice with our Cre mice. So here Cre is under control of the insulin promoter. So when this promoter is active, which is only in beta cells, Cre is expressed and then recognize ATF6 to knock it out. So that's how we get our deletion in beta cells only. The bottom two panels are the knockout confirmations. The left is at the RNA level through qPCR, and this is for whole islets. And at the right is at the protein level with IF. So here you can see that the controls have ATF6 expression, but it's severely diminished in our knockout model. So we followed these mice from 3 weeks all the way up to 50 weeks of age and measured their blood glucose. And on the y-axis here is the diabetes incidence. And we found that, interestingly, these knockout mice were more protected. So the flux and pre-only control were around 55 to 65% diabetic. While the knockout-
Hugo, I just wanted to
Yes.
bring to your attention. I think we can hear you better when you're closer to your microphone.
Oh, I'm sorry.
No worries.
Okay, I'll stay close. So the flux and pre-only control were 55 to 65% diabetic, while the knockouts were only around 50%. So to look at what's happening at the tissue level, we looked at immune infiltration in these mice. So here at 5 weeks of age, with the H&E, you can see healthy and normal-looking islets without any immune infiltration. But at 10 weeks of age, the controls, you see these dark and dense dots, the immune cells infiltrating and attacking the islets, but our knockouts are protected. And when we quantify this, we see that knockouts have more infiltration in both aggressive and non-aggressive types, and also more intact islets, which are islets without immune cells.
Hugo,
Uh,
can you take a question? We've got a question in the audience. Did you generate the insulin 2 promoter Cree ERT mouse yourself, or did you use existing JAX mice?
This is the Doug Melton cream mice that we backcrossed into an OD background, but I believe it's also available in JAX now if you want to get it.
Great, thank you.
Okay, since we saw much less insulitis in our knockout mice, we then looked at cell death by staining for Nexin-5, which is an apoptosis marker. And we found that knockout cells have reduced cell death, which is consistent with our diabetes protection phenotype. So far, we observed that our beta cell knockout of ATF6 ultimately protects them from type 1 diabetes. But we wanted to ask, what's the mechanism of action at play here? So to answer this question, we performed RNA-seq on sorted beta cells. So here you can see the top regulated pathways, and you might notice that the top one is p53 signaling, and this is important for tumor suppression, but it's also important for this pathway called cellular senescence. So that's where we shifted our focus to next. So senescence is an adaptive response. So senescent cells undergo a cell cycle arrest and release a cytokine cocktail called SASP that can recruit immune cells. And in healthy settings, these immune cells can clear the senescent cells away. But in some cases, like in aging, where immune cells are impaired, the senescent cells can't be cleared, and you get an accumulation of SASP and terminal senescence. So while transient senescence can be beneficial, persistent senescence is detrimental. And these are also some of the markers of senescence. You can have increased levels of P21, P16, DNA damage response, cell cycle arrest, increased cell size, and also more senescence-associated beta-gal activity. So we measure these as senescence markers, and we'll see more of these guys later. So the next thing we did was look at pathways that are affected during senescence. So here you have the p53-phenolphthalein pathway, the antioxidant response, and DDR. And we saw that these genes were differentially expressed in our knockout model. So that's the mRNA level. So we then looked at the protein levels of these senescence markers with IF. So the first one is P21, an important marker. So what we found was that at 5 and 10 weeks of age, significantly increased, which is consistent with what we saw with our RNA-seq. But 20 weeks, it wasn't significant anymore. And phosphorylation of H2AX is also a marker for DDR, which is a marker for senescence. And here we see that there's also increased in our knockout ATF6 mice, similar to P21 in the last slide. Then to look at the cell cycle, we did DNA content with flow, and we can classify them based on like which phase of the cell cycle they're in. And we found that at an earlier time point of 6 weeks, the ATF6 knockouts were more likely to be stuck in this G2M phase. And when we were trying to figure out the mechanism for our model, this paper came out which showed that P21 activates a secretory phenotype called PASP, which is different from SASP. but it can also recruit immune cells. So we wanted to see if it played a role in our model. So we compared the pass factors in their paper to our models, and we determined some core factors. And what caught our eye was that several macrophage attractants was increased, suggesting that macrophage recruitment might be affected in our model. So depending on its activation, macrophages can polarize into different subtypes. So there's the M1 macrophages, which are considered classically activated and inflammatory. And there are also the M2 macrophages, which are anti-inflammatory. And actually, it was found that Doppler transfer of these M2 macrophages to NOD mice can prevent type 1 diabetes. And since our model also has a protection phenotype, we decided to look for M2 macrophages next. And so we stained for arginase, which is an M2 macrophage marker. And we found that in our knockout model, there was increased M2 infiltration into the islets of our knockout mice, as you can see over here. So there's increased recruitment into the islet. So you can see in here, the knockout have a greater number of M2, and they're also more infiltrating into the islet itself. So we also looked at polarization in the whole pancreas, where we're seeing for surface markers of these macrophages. And I found that decreased percentages of the M1 population and increase in the M2 population. So macrophage polarization as a whole is also affected in the pancreas, in addition to the recruitment to the islets. So we then wanted to see whether or not this macrophage recruitment is dependent on this P21 pass in the knockout cells. So in order to test this, we use a beta cell specific knockdown of P21 at 3 weeks of age, and then collected their pancreas at 5 weeks. And what we found was that this knockdown decreased M2 macrophages in the islets, showing the importance of P21 and also PASP in this macrophage recruitment. So next, we wanted to see which PASP factors play a possible role in the M2 recruitment. So I previously showed this figure where a gene called LIF is increased in the knockout mice. So to see if LIF was important in this recruitment, we treated the mice with either IgG antibody for control or an anti-LIF antibody to neutralize it. So here we found that it decreased M2 macrophage infiltration, showing that LIF is an important part of the PASP and important for M2 recruitment. And to tie the two together, we also show that LIF is dependent on P21. So this is a Western blot using using a cell line. So to mimic our ATF6 knockout, we used Sayapin, which is an ATF6 inhibitor. And you can see here in the computation that when ATF6 is inhibited, you have increased LIF, as we would expect, and consistent with our in vivo model. But when you inhibit P21, LIF goes down. So LIF expression is dependent on P21. So just to summarize the section, we know that our knockout mice have increased adaptive senescence, and P21 plays an important part in here, which leads to the secretion of P21 PASP, including LIF, and this leads to the M2 macrophage recruitment to the islet. But our results with increased early senescence in contrast with a previous paper. So it shows that if you remove the terminally senescent beta cells in the islet, you can prevent type 1 diabetes. So here you can see in disease progression, some beta cells become terminally senescent, but if you remove it with a drug, you can preserve beta cell mass and prevent type 1 diabetes. So we wanted to see if this late terminal senescence have a role in our model. So we previously looked at cyclical rest at 6 weeks of age and saw a significant increase in the population in the G2M phase. But at 12 and 16 weeks of age, the difference is less apparent. And at 16 weeks of age, it's not even significant anymore. So this shows that either the cells that are stuck in the G2M phase over here at early time point 6 weeks are either exiting the cell cycle or being cleared. Another marker for late senescence is the beta-gal activity. And we determine the activity by staining with C12FDG. And beta-gal can cleave this to create a fluorescent product, which we can detect with flow. So at 5 weeks of age, early time point, we don't see a difference, as you can see over here. But at 20 weeks, later time point, ATF6 knockout mice have reduced senescence-associated beta-gal activity, indicating less late senescence. And the increased cell size is another hallmark of late senescence. So we see a similar result here as well. At 5 weeks, early time point, no difference. But at 20 weeks, control cells are larger, but knockout cells stay relatively the same size, so, which is consistent with our previous data. So overall, our ATF6 knockout mice is showing reduced terminal senescence at later time points. So from here, I just wanted to pivot a bit. So far we focused on our ATF6 mice over here, but we also have a different mouse model in the lab, which is our RE1 knockout model. And this is also a knockout in beta cell only. So RE1 is another sensor of the UPR, and it's an interesting one because it has both kinase and RNase function. So as a kinase, it can engage various inflammatory signals, which can lead to apoptosis. And as an RNase, it can activate a couple of pathways. One of them is RID, which is responsible for degradation of mRNA. And it can also splice XBP1 from its unspliced form to a spliced active form, which is then translated to a transcription factor, which can also regulate UPR target genes. So in our RE1 mouse model, we found that they have a similar phenotype. And I'll just go over it a bit quickly since we already saw the same kind of results before. But here you can see that in the 6 weeks cell cycle analysis, the RE1 knockouts are also more likely to be stuck in G2M phase. And they also have increased P21 beta cells, along with increased phosphorylation of H2X, which is what we saw earlier as in ATF6 mice. And they also have increased macrophage recruitment to the islets, same with what we observed in the ATF6 mice. So here you have the number of arginase cells in the eyelid. And this is the polarization in the pancreas. You can see that they also have decreased M1 polarization and increased M2 polarization. And they also have reduced terminal senescence. They also have reduced C12FDG activity, as you can see over here. And they also have reduced size at 20 weeks, just the later time points. So we also validated these results in human islets. So these are different PAS genes at the bottom over here. And the red bars, or the red bars over here, were inhibiting ATF6. And this blue bar, we're inhibiting IRE1. And you can see that compared to the control, inhibition of ATF6 and IRE1 results in increased expression of these PAS. And we also use the HPAP data for this. So in type 1 diabetes, so not all the beta cells are gone. There's also residual beta cells. So we looked at their P21 pathways and path genes in these residual beta cells of type 1 diabetes, and we found that it was increased. And since path had a predictive effect in our knockout model, this might be contributing to how the residual beta cells were able to survive in this disease.
Hugo, can you comment a little bit about the or the phenotype of the residual beta cells?
This is for this one. This is using the HPAP data, the single cell RNA-seq data sets that are publicly available. So we didn't analyze the morphology itself for these beta cells.
So you just went into the data?
Yes.
Yeah, human data.
Yes.
Okay, great. And then there was no like discernment between whether, what was any kind of characteristics of those beta cells that were the residual.
They were classified as beta cells due to their insulin expression. So we know that they're surviving, but
just it's interesting because scientists like Guy Rutter and, others, kind of point to the fact that some beta cells could be potentially pacemakers and others, maybe not. And it would be interesting to understand, what's happening to these past genes in the residual beta cells. What are those beta cells that are residual, I guess, in this context? But yeah, maybe you can't figure that out from these
Are
Yeah,
data.
we
you
can
asking whether these are hub cells or leader cells or subspecification of the beta cells, and whether we see this past phenotype with the distinct subcluster of beta cells?
Exactly. Hmm.
Yeah, we have not gone that deep yet, but that is something that we're working on actually.
Yeah, because it's really neat. This is some really interesting data. Okay, thank you so
Okay, thank
much.
you. So to summarize, on the left is the normal NOD mice. So you can see it develops immune infiltration and develops diabetes. And in contrast are ATF6 and RE1 mice secrete PAS factors such as LIF, which includes which leads to M2 macrophage recruitment. And this leads to reduced terminal senescence and reduced diabetes and healthier islet at later time points. And we also showed, using the publicly available data from HPAP, that the humans also have a similar pattern. So right now, we'd like to acknowledge the current past members of the lab, which including Samra, who was a co-author on this paper as well, and also the collaborators on the left side here who was working with us on this project.
Great.
Yeah,
Yeah, I
thank you.
see. Yeah, thank you. I see that Decio is involved. No surprise, and that's great.
Yeah, he
Given
was helping us with the human islets.
right, and he's got some nice review papers, historical review papers regarding the UPR in context of beta cells. Just want to open it up to people if they would like to drop a question in the chat. We can ask the group, or you can unmute yourself and ask the question directly. Please feel free to do so. I wondered if you have sort of what's next for you, Hugo.
What's next for me? Like
Mhm.
after actually today is my last day in the lab, so
Oh,
I'm
that's bittersweet.
yeah, I know. It's the last presentation, so I'm just waiting to hear from a couple of jobs in industry. So we'll see what happens from there.
Okay. Well, fantastic. I'm hoping selfishly that the industry is diabetes related, maybe. Um,
We hope so too.
yeah. And I don't know, Feyza, do you want to comment on sort of maybe a little bit of other projects that are ongoing in the lab that you feel comfortable sharing?
Yeah. So historically, we're working on beta cell stress in type 1 diabetes since 2008, during my postdoctoral years. And following our initial paper, my postdoctoral work, we really wanted to understand specifically the ER stress pathway. But those studies are really branching out to different angles. As you see now, we are sort of really interested in understanding the early senescence, because now we have very unique models that we can ask very specific questions.
Yeah.
This is the first time that I think that one of the important the impact of this paper is that it's the first time showing that beta cells are doing immune surveillance when they're stressed. And then there is a clearance mechanisms. And this is just like the first paper, but now that we have these models, definitely we would like to understand the characteristics of these early senescent beta cells, how they are behaving, what is their antigen responses, how do they communicate with immune cells, whether other immune cells are also involved in this immune surveillance mechanisms, and how, if is it possible that these mechanisms are going are impaired during type 1 diabetes process, and can we figure this out? Can we find ways to induce the immune surveillance in an optimal way that we can actually reduce the accumulation of terminal beta cells? So definitely this paper is just like the first, but we have great models to ask these questions.
Yeah, you
And
really
then
have. You've established a really elegant model in a very complicated pathway or set of pathways, parallel pathways. Great.
pathways, exactly. But there's also the metabolism part of it, organelle dysfunction of it, which are also involved in senescence and ER stress. We have some projects going on in the lab. Organelle crosstalk, ER mitochondria crosstalk, ER lysosomes, and sulfate determination and the role of metabolism on it. That's another project that is going on in the lab. Another stress pathways. So it's really how we can use this body's natural stress responses to help beta cells survive the immune attack as one of our long-term focuses for the lab. But yeah, there's the multiple angles. But these are very valuable models that we like to do more work and go deep. Like one of the questions that you asked, right? Really specifics of what kind of beta cells are, because beta cells are very heterogeneous, as you pointed out. Which cells are showing this? Are they specifically targeted? Are they differentially targeted? All those questions we're interested in tackling.
Yeah. I mean, if you think about it, it's so interesting to think about the idea of the pacemaker beta cells in context of the human islet, and whether or not they are targeted first as they undergo increase UPR. Or do they undergo I mean, I guess my question would be, do they undergo UPR um stress-induced UPR first? Are they first offline? Or does it seem like maybe the other beta cell phenotypes go offline first? What lights get shut off first, I guess, is sort of an interesting question.
All right, I think we are in a really great time to tackle these questions. There's a lot of bioinformatics that we can use. Trajectory analysis, time course analysis in the case of human patients, like from autoantibody positive couple one autoantibody to multiple new onset type 1 diabetes. Samples are available through the long-term diabetic samples. So if we use the current knowledge and technology to track these cells, at least bioinformatically, to see what's going on, I think we can get some information. But we always go back and forth between human and mouse. The same things that can be done in NOD mice too, go back to humans. But definitely we're in a better place compared to like 20 years ago to ask these important questions.
Yeah, no, I think the advent of of big data is going to really inform a lot of these quests. So it's fantastic. And I wanted to also, your lab is very highly regarded. You guys have a lot of really interesting and exciting work coming out. Are you looking for graduate students and postdocs?
Always.
Okay.
Whoever is passionate to do this kind of work, we're always waiting to recruit these trainees and ask these questions together, tackle them together.
Yeah, I would really encourage anyone to pursue this, to reach out to you and pursue this avenue of thought. I mean, you've got some of the really great collaborations between the Thompson Lab, Zurich Lab, Anil Bhushan's lab. It's really, really sort of cutting edge thinking about how these things work. So just really congratulations to Hugo, and thank you both for joining us. I hope you have a great exit party today, Hugo. And if we hear of anyone also interested in this type of work, we'll send them your way. Just a really nice really nice paper and really great work.
Thank you so much.
Thank you.
Thank you again. Oh,
sorry. I have two questions if Dr. Engin has some time.
Oh, sure.
Hi, my name is Maura Doyle, and I work with Dr. Egan over in the NIH, and we've been looking at the CB1 receptor in preventing inflammation in islets in type 2 diabetes, and we're now looking at it in type 1 diabetes. So my first question was related to the earlier work on type 2 diabetes, looking at senescence, and the earlier papers showed that senescence really had no effect on the progression of type 2 diabetes. Whereas your work indicates that it does seem to have an effect in type 1 diabetes. So do you care to comment on the type of senescence that's occurring in type 2 versus type 1? Is there any relationship to, say, a difference in the type of infiltrating immune cells that could be affecting the type of senescence that's occurring in the islets? And then my second question is, I just quickly looked at your paper in Press. The mice that you're using, as Dr. Lee said, are coming from Doug Melton's lab. Are they tamoxifen inducible? And how does that affect how you treat the NOD mice in terms of the progression of their diabetes? I'm just curious as to the mechanics of that, if you wouldn't mind, please. Thank you.
Thank you. I'll start with the second one. That's what I remember easily. So we use, yes, the Cree line that we use is tamoxifen inducible. And the way we induced the deletion, it was because we wanted to delete these UPR sensors during the early stages of diabetes. Even before the autoimmunity starts, we injected the lactating mothers with tamoxifen, meaning that both the control groups and the knockout groups receive the same amount of tamoxifen, which also allowed us to delete these genes early in the disease progression. Does this answer your question?
Yes, thank you. So does injecting in the lactating mothers, that will knock it out then, will that effect last longer than 6 weeks? Because I do know the effect of the tamoxifen wears off after some time. Are you looking at them beyond 6 weeks?
So we actually did this experiment to see whether when you knock out, for example, we did it for IRE1 when we deleted IRE1, and then we collected islets at 16 weeks of age to see whether there is like overcompensation and proliferation of the other IRE1 non-deleted cells or so. But we can actually see that IRE1 is still significantly diminished in beta cells. We use Ziegler to sort beta cells to do this Western blot to see whether the IRE1 is still depleted, and it was there.
Great, thank you so much.
So in terms of the type 2 versus type 1, so actually I should say that the initial paper is from Peter Thompson's lab, where he showed, briefly, Hugo also showed that these senescent cells accumulate with time in at least in the type 1 diabetes model NOD mice, and a pharmacological depletion of these cells show a beneficial effect in type 1 diabetes. But we should also look at this, what kind of senescent beta cells are there? Well, P16 positive, P21 positive, they have their own characteristics. With type 2 and type 1, that might be the differences, what kind of senescent cells are accumulating. And every cell type is having their own unique senescent cells and their unique SASP, unique PASP. And I think it wouldn't be surprising to see differences in the context of type 2 versus type 1 because of the different inflammatory milieu this tissue environment has. But I think we're still just like on the very tip of this these questions that we will need more data to compare what's going on in, let's say, for beta cells in type 2 versus type 1 diabetes context.
Thank you. Thank you very much. It's very interesting work, and I've been following your work and your reviews on UPR in the beta cell for a number of years, and they certainly have guided what we have been looking at in our models. Thanks very much.
Thank you so much.
Great
I see-
questions. We have one more question, yes, from San Diego, Matthew Wortham, and here it is. Do you know the fate of early senescent beta cells? Is there evidence that they can revert back to non-senescent beta cells? And also, is there an assumption that they can progress to late senescence, or are these distinct cell fates?
Yeah, that's a great question. Nice question. I wish I knew the answer, but I can probably speculate. Something that we will be very interested in what is going on, because the best way is to answer this question to have some sort of lineage tracing that we could actually find this P21 expressing early senescent cells and see what's going on. Are they converting to P16 expressing senescent beta cells, or what percentage are being cleared? If they're cleared, is there like sort of switching to another permanent type or terminal senescent beta cells, that these early senescents are becoming terminally senescent beta cells? It's hard to answer it with the current experiments that we're doing. Again, if there was a way to do lineage tracing, and the hard part is that even if you have some ways to do it, you need to put it onto an OD background if you have a reporter or so. And that takes forever, unfortunately. Even with the speed congenics, it takes forever. And for these early beta cells, another issue to answer this clearly is that, so these are flux mice together with Cree. And if we introduce another way to track them, getting the animals that has all these genes, all these alleles, the rate is like probably 1 in, I don't know, 6,400 or so. It's tricky experiments, but I think the questions are really interesting. Maybe we will also use some trajectory analysis, do some bioinformatics. Biologically, genetically, it's a little bit hard to tackle because of this background issues. But it's a very important and nice question. Thank you.
Yeah, I
I
mean,
appreciate it.
oh, sorry.
Thank you. I appreciate that. Yeah, I understand. Yeah, I think I completely agree. It'd be really interesting. I wonder if there are culture models of some reversibility that could be used. But yeah, it's great. I really appreciate it. And I enjoyed the talk and reading the paper as well. Thank you.
Thank you for attending. For cell culture, I mean, we did some cell culture studies, and some of it we can mimic in NIT1 cells when we pharmacologically inhibit either ETF6 or IAD1. We can recapitulate some of the phenotype that we, not exactly 100%, but some we do. Actually, the initial study is this all macrophage migration. It all came from the in vitro studies. That it's macrophage specific. It's not the lymphocytes and other, but I prefer in vivo. If we can do this, I would feel more comfortable to find a way to answer these questions in vivo. But I am very interested in these questions, obviously.
Yeah, no, it's very, those are great questions. Just one sort of offshoot question. I know we sort of ended and then went into questions, but you know, when you talk about the sort of the difficulties of these model systems, sometimes in developing these sophisticated, you know, Cree mice or mice models, has anyone looked at this paradigm in zebrafish? I know Teresa Mastracci is very involved in, you know, zebrafish model systems, looking at how the pancreas develops in type 1 diabetes and so forth. But is there any kind of model that goes on in zebrafish that's comparable, or no?
There's a paper in Cell Reports last year, I believe. It might be Vanderbilt, or so I don't want to say something.
Yeah. But they have looked. They have looked. So
yeah, they use zebrafish model, and then they were showing that there was this CXCL5, or I don't remember, chemokine that is sort of helping recruiting immune cells and clearing stress beta cells. That's the zebrafish model. I think it's an interesting paper. But yeah, it's hard to tackle some of these questions with NOD mice due to the background issue that you're not as flexible as working with the Black 6 mice, that you can just breed so many alleles and track them. But we'll do whatever we can and to answer some of those questions, because I think scientifically these are really interesting questions, and in the long run we need to know the specifics of these characteristics of these beta cells to develop very specific therapeutics that can be used.
Yeah, it's a bad type 1 diabetes is sort of a bad ballet. You know, it's not like one and done. Like you can't just see, oh, here's what happened. So I appreciate your efforts in trying to really fully understand this sort of bad ballet. But thank you all again, and everyone for their really insightful and interesting questions. And I hope you have a great rest of your day and a nice holiday season.
Thank you for joining us too, and thank you for the invitation. Thank you very much.
Thanks again. Bye-bye.
Bye.